Hey everybody, my name is Mason Hansey and this is MHC and immunoglobulin diversity. So overview, proteins in the MHC complex, genes that do coding for MHC molecules found on cell in the body. Split into two classes, MHC1 and MHC2, both represent glycoproteins that are also dimers. MH1 molecules are composed of a long protein chain with a macroglobin protein coupled to it. MHC2 molecules consist of two proteins chains labeled and coupled as A1, A2, and B1, B2. MHC class 1 genes have three genes coding for polymorphic MHC class 1 molecules. This would be HLA-A, HLA-B, and HLA-C. Three additional, HLA-E, HLA-F, HLA-G. These are structurally like polymorphic C1 genes. HLA-E and HLA-F present antigens to NK cells. Here we also see MICA and MICB genes. The codes that these genes deal with are those that interact with NK and T cells. They are not found on the surface of a cell. MHC class 2 genes. These are genes that code for polymorphic MHC class 2 molecules like HLA-DP, HLA-DQ, and HLA-DR. They contain an A and B gene, can also contain DRB genes, possible to have 6 class 2 MHC molecules on the cell. This includes HLA-DM, this codes the molecule that deals with the peptide exchange in the MHC2 pathway. MHC class 2 consists of alpha and beta chains. Polygyny MHC haplotype. This is where the MHC class 1 and 2 molecules are coded using different genes. HLA-A, B, and C code for class 1 MHC molecules. 6 different molecules can be expressed that can be binding with peptides. This same complex can also code for MHC class 2 molecules that consist of HLA-DP, HLA-DQ, and HLA-DR. And you can see that picture to the right. And that's where it's talking about the single characteristic based with the several different genes or molecules that can control that. Polymorphism and the MHC haplotype. This is where multiple stable forms of the MHC gene can exist in a population. The MHC is the most polymorphic gene in the human body. Differentiation of RBC antigens is an example of polymorphism in humans. This varies quite drastically between each human. Overall, it's very diverse, but the same set of class 1 and class 2 molecules is the same in every cell in the body. Regulation of MHC molecules. Class 1 and 2 are codominantly expressed. Cell expresses MHC molecules transcribed in paternal and maternal chromosomes. Can also be coordinately regulated. This means all class 1 and class 2 molecules are expressed simultaneously on the same cell, regulated separately due to the expression of MHC class 1 in the absence of class 2 molecules. Inheritance of MHC genes. Haplotype is passed to the offspring. The HLA genes will differ due to the variety found in parents. This is due to the chances of two parents that are of different lineage won't carry the same combination of MHC molecules. These haplotypes differ between each person in a family. They have the same base but don't carry the exact sequence. Immunoglobulins and their genes consist of light and heavy chains. The heavy chains include the IgM, IgG, IgD, IgA, and IgE isotopes. VDG or J segments code for the variable region in the heavy chain. Heavy chains can undergo VDJ rearrangements. Light chains, smaller than the heavy chains, and have VJC segments. Somatic hypermutation and affinity maturation. Somatic hypermutation, these are random point mutations in the VDJ unit of antibody H or L chain V region genes. This mutation can change amino acids, facilitates diversity. Affinity maturation, this is the further development of the B cell in response to antigens in lymphoid organs, generates antibodies with increased binding affinities. And these are my references. Thank you for listening.